Hey everybody, so my name is Jesse, and I'm here to talk about single cell sequencing analysis in R, specifically using Seurat. Um, so prior to this video and prior to this section, you have already heard Chris talk about using Cell Ranger to process FASTQ files from a single cell sequencing experiment. And Cell Ranger will do the mapping and the alignment and produce a count matrix where where each row is a single gene and each column is a single cell. And then the data within that count matrix are the number of reads detected in a single cell that align to a particular gene. And so this count matrix is essentially what we're going to use to generate objects in Seurat, where Seurat, we're going to do normalization, scaling, and eventually dimensional reduction. And then within Seurat, we can also do clustering. So we're going to generate clusters and visualize these clusters and use these clusters to do differential gene expression analysis. And then beyond Seurat, if we want to do more after that, we'll have to use other programs, which Hope and Georgia will talk about, and Mary will talk about after this. So Seurat in general is a pretty widely used R package for generating or for analyzing single cell sequencing experiments. I'd even say it's pretty much the gold standard of doing single cell sequencing analysis. I think maybe you'll see some people do these types of analysis in packages in Python, but in general, people are doing this in R using Seurat. So again, Seurat allows you to do everything from the initial quality control where we're removing debris, doublets, all the way through the normalization and scaling and dimensional reduction of our data. I always find this really interesting that Seurat was named after a painting from the 1800s called A Sunday Afternoon on the Island of Le Grand Jatte, which is an impressionist painting. And in the paper that published this package, they acknowledge that they named this package because of the similarities between a UMAP dimensional reduction plot as shown here, or sometimes we'll know this more colloquially as cell clustering, but there's a lot of similarity between the way these plots look and early impressionist paintings, pointillistic paintings. And so, yeah, I'm going to walk you through how we process these individual Seurat objects, how we're adding metadata so that when we plot the data, when we plot the cells, we can then go back and visualize which cells came from which experiment. We're going to find variable genes and do differential gene expression. And then we're going to integrate the Seurat objects across different experiments, across different technical and biological replicates, in order to reduce the effects of different batches and different experimental variables. And then we're going to do the plotting and the visualization. Okay, so now I'm going to walk you through the code, and this is the worksheet that we're going to be working on. So the first thing that we're going to do when we are working in R on this cluster, we're going to make sure that we set the R version correctly to R 4.40. And this is really important because all of these packages are loaded specifically into this particular R version. If you are selected on any of these other R versions, you will not be able to see it. You will not be able to load in any of these packages. Let me see. Right? Okay, so once we've loaded it, once we've ensured that we're in the right R version, we're going to load in the packages that we're going to need. The first one obviously is Seurat. This is already all installed into this R environment. So all we have to do is type library, Seurat. We're also going to use tidyr and dipler. And these are just packages that are useful for looking at our data matrices, and it provides a more updated version relative to the base environment that allows us to really efficiently visualize data tables and data frames. Okay, so the first thing we're going to do is I like to set a working directory. This just makes my life easier as we move forward, so I don't have to keep typing in the same path. This is where all these files that come out of Cell Ranger are going to be located. So Cell Ranger will produce a plethora of files and directories. The ones we are going to use for this particular version of reading in these files is going to be in the outs directory. and then in this filtered feature underscore BC or barcode matrix. And so there are a lot of different ways of reading in output from something like Cell Ranger into R to generate a Seurat object. I like to use this read 10x function. It makes things really easy. You can also just take the count matrix itself, the raw count matrix, and generate a Seurat object that way. This is a lot more efficient, I find. And within this directory are actually going to be three different files. There's a barcode matrix, a features matrix, and something called a matrix.tsv or .matrix file. And these files together are what this read.10x or read10x function uses to create a Seurat object. And so that we're literally just directing it to the to this whole directory. And so within the we have named each of these Cell Ranger output directories differently. So I'm just pasting the unique name that was given hopefully by Chris, and then I'm gonna direct this read10x into this subdirectory. And then we're going to do this create Seurat object, and this is part of the Seurat package. Well, so is read10x, but this is just a way of generating. So what comes out of here is more of a raw file, and now we're actually going to create our Seurat object, and then telling it what to call the count matrix and what to call this project. It doesn't really matter what we call the project because we're going to add metadata separately. So once we've read in all of our files, we have, in this case, six different Seurat objects. And now we're going to add metadata, and adding metadata is super important. And we do this really early on so that all these individual Seurat objects will have this metadata assignments, designations as we move forward. We can add as much metadata as we want. We never can have too much metadata. Really, so adding metadata is really simple, just like adding a column to a data frame. We're just going to type in the Seurat object, and then we're going to direct. So the way that the Seurat objects work is that there are nested data frames within each object. And the way we get into this nested structure is using this @ symbol. So we're going to go to the metadata nest within this Seurat object, and then we're going to add a metadata column called gender. And it's really simple. We just add the $ to metadata, and we can call this whatever we want. And then we assign it based on the information we know from our experiment. So I'm assigning female to all the females in this experiment and males to all the males. I'm also going to add T21 status. So again, I can name this whatever I want, but I'm just going to add assignments as to whether a Seurat object is corresponding to a trisomic or a disomic experiment. And that way, within each of this object, I can look at metadata and I can say, I'll be able to access both the T21 status and the gender of every single object. So I'm able to understand the relationship between these multiple variables as we move forward. Okay, and now once we've added metadata, the common next step is to do a quality control. And so the most common form of contamination in single cell RNA sequencing is doublets. So that's when two individual cells will end up in the same liquid droplet using the 10x genomics machine microfluidics device. And if two cells end up in the same droplet, they'll be labeled with the same UMI. And so they'll look like a single cell, even though they're multiple cells. And so one really quick and easy way of dealing with this, prior to that point, we're going to, prior to looking at doublets, another common contamination is mitochondria being labeled as a single cell. And so mitochondria are like cells are membrane bound, and it's common that a mitochondria can end up in a droplet and look like a single cell. But when the genes from that mitochondria are mapped, 10x or the Cell Ranger suite will actually assign each of those genes with a specific pattern. So this MT dash gets assigned to all genes that are from the mitochondrial genome. And so I'm able to set a percentage feature set. Essentially, I'm generating basically a piece of information that captures what percentage of genes that mapped to the mitochondrial genome are present in each Seurat object. And that's what I'm doing here. When when we do this, you'll be able to do a violin plot, and you'll be able to look at, for each cell, what is the percent of mitochondrial genes that are mapping to the mitochondrial genome. We're able to look at the amount of genes that are mapped from an individual cell and the amount of reads that were captured from an individual cell. And so we're going to use these percentage sets, these distributions of genes and counts and mitochondrial contamination or mitochondrial genes to filter cells that we think are either doublets, are cell debris, and so cells that maybe broke apart and really what we labeled was just loose floating RNA and not necessarily a distinct single cell, or a mitochondria rather than an actual cell. And so what we can do is we're actually, for each of these Seurat objects, we can subset based on cutoffs that we can assign. These cutoffs are the cutoffs that are recommended by the Seurat tutorials that you can find online. But depending on the way these distributions look, we can actually change these cutoffs accordingly. But for now, we're just going with the standard thresholds that they recommend. And so I'm basically, in these lines, I'm filtering out all cells that have less than 200 genes mapped to an individual cell. I'm filtering out all cells that have greater than 2,500 genes mapped to a single cell, and I'm filtering out all cells that have more than 5% mitochondrial genes being mapped to it. So I'm filtering out all cells with very few genes. So these are likely debris. We'd expect a single cell to be expressing more than 200 genes. So anything less than that is probably not a single cell. I'm filtering out all cells that are expressing more than 2,500 genes because we don't expect a cell to be expressing that many genes. And if they are, they're probably not a single cell, but rather a doublet. And again, I'm filtering out cells that have a high percentage of their genes being mapped to the mitochondrial genome. But really, we can adjust these thresholds based on the way the distribution looks. Oftentimes, we'll see a sort of bimodal distribution where it's really obvious that there is a distinct population of doublets or a distinct population of mitochondria, that maybe this is not 5%, but maybe this is 10% or 1%. Maybe this is not 2,500 genes where we see a distinct population of doublets, but maybe it's 3,000 or maybe it's 2,000. So this is a tunable step where we're able to adjust our thresholds accordingly. But I usually stick with these recommended values because they're justifiable based on the work that went into developing this program and by the group that published this. So the next thing we're going to do is do a integration. And so there are a couple different ways of doing this. Integrating the data is really important because it's going to correct for batch effects. So correct for technical variability across experiments. And this is really important because every time you do a single cell experiment, there are all kinds of, it's a very sensitive experiment, and there are lots of different ways in which individual cell preparations and individual runs on the microfluidics or on a sequencing run can produce all kinds of technical variation that we want to correct for. And so prior to actually correcting for these differences, the first thing that we're going to do is to do what I call a simple merge, which is just using merge, which is part of the base R environment. I'm basically just creating a list of all these different Seurat objects so that when I go through these next steps, I can do them all as one instead of doing the next steps individually on each Seurat object. So after I merge, this merged object is essentially just a list of all the Seurat objects. I'm then going to do these 4 functions that are pretty that are inherent, that are really critical part of this Seurat workflow. The first thing we're going to do is normalize the count matrix, and then we're going to find variable features for each of these. So I'm basically, even though I'm running this on the merge, I'm on the list, I'm normalizing each Seurat object individually, although it looks like it's all happening to as a whole. It's really happening to each object individually. So I'm going to normalize the count matrix for each of these objects, and then I'm going to do this find variable features. And essentially what that's doing is it's finding genes that are highly variable across cells, as opposed to a gene like a housekeeping gene that may not show a lot of variability across different cell populations. And so it's using these variable features to to eventually generate clustering, to generate the clusters and do all downstream analysis, as opposed to using all genes, where really there's a lot of genes that we don't care about, genes that are going to be ubiquitously expressed across cell types, because ultimately we want to identify different cell types. And so we're going to look for the genes that show high variability across different populations. And then we're going to scale these count matrices and then run a principal component analysis. So PCA is a linear dimensional reduction technique, and it's important for although we're not going to use this to visualize our data, it's an important step for the later non-linear dimensional reduction, which is UMAP. But so once we've gotten through these additional dimensional reduction step, then I'm going to do this integrate layers function. And there is so in this case, I'm using this RPCA algorithm. RPCA stands for reciprocal principal component analysis, or reciprocal PCA. There are, I think, 4 or 5 different functions you can use, algorithms you can use to do this integration. If you want to learn more about them, there's a lot of information about the math that goes into this on the tutorial. But I found that this RPCA algorithm usually works well with my data sets, and so that's just what we're going to use today. And yeah, and that's the end of this first day. And tomorrow we will get into the actual UMAP clustering, the dimensional reduction, in the initial attempts at identifying different cell types, doing differential expression across different cell populations. And then after that, you'll go into a more in-depth analysis.